midterm exam, so if you would like me to look at your exam, please drop it off at the end of class. And there are a few sad, unrecovered midterm ones up here, for those of you who haven't picked them up yet, although I suspect that everybody here has picked them up. Okay, so today is our third and final lecture on regulation. Next week we will be doing some material on nitrogen metabolism and on fats. And then we're into the second midterm, right? The following week there's a review session. There's no quiz actually due next week. There's a quiz due the following Monday. The next quiz, which will be online early next week. And then the midterm is next Wednesday. Is that accurate? Yes. Note that the midterm covers all of Chapter 14, even though the beginning part of Chapter 14 was already covered on midterm 1. That stuff's also on midterm 2. It's the glycolysis pathway, so it's pretty much impossible to separate it out from the rest of the material we've covered since then. There's another sample midterm from last year is online, and the answers. Some other materials are online for next week, and chapter problem answers and such. Okay, so today we're going to talk a little bit more about the individual regulation of enzymes in glycolysis, really just the fructose bisphosphatase, the PFK1 step, and the FBPH1 step in gluconeogenesis going the other way, which is what we started talking about last time. And then we'll get into the regulation of glycogen synthesis, which is a little bit more complicated in that it involves more hormonal control. So this third enzyme, I think this is actually one of the reasons I think why there was surprise around the results of that metabolism experiment, where extra amounts of the enzymes were titrated in, and the hexokinase had the biggest effect as compared to phosphofructokinase 1 in that metabolic control analysis slide we talked about last time. Probably because there's a lot more regulation going on at PFK1 in the sense that there's more types of regulators and there's more levels of regulation. Right? I mean, we talked about the regulation of hexokinase, certainly significant, apparently very significant if you believe that control analysis experiment. But a lot is going on PFK1 for sure. So this map is showing you, for that step, the interconversion between F6P and F16BP, where you can be going in either direction in two pathways. You have dedicated enzymes. One of them is working at any given time, but both of them are not working. We talked about why. We just write down the two reactions and add them up. The sum of those two reactions is ATP goes to ADP plus phosphate plus heat. So that would be the Butyl cycle if they were both running at the same time. ATP and citrate inhibit PFK1 because they signal high energy states of the cell. Right? And so we don't need to be burning more glucose to generate more ATP if we've already got a lot of it. So we ought to instead be running gluconeogenesis in a situation like that. If we have a lot of ADP or AMP, that signals low energy state, so yes, we do need glycolysis. You see here that AMP both activates PFK1 and inhibits the gluconeogenesis enzyme. So if we have a low energy charge, we certainly want to be enhancing gluconeogenesis. We want to be blocking gluconeogenesis because we need energy to do it. So AMP is effective first because it's operating in both enzymes. It's an inhibitor that's operating on both enzymes, so it will go one way or the other. And it's also effective because it's at a lower concentration, so changes in its concentration are sensed more easily by enzymes that bind it because they have bigger percentage effects on the total concentration, so they move you up and down the B versus S plot a lot more. Right? So we talked about that in the context of that other enzyme, AMP-dependent protein kinase, that phosphorylates a lot of enzymes in lots of different tissues. Okay. So the additional level of control is this F26BP metabolite, right? And so fructose 2,6-bisphosphate, you can see it here on the bottom. Here's the structure of it. It's just bisphosphorylated fructose sugar. And the way it works is that if it's around, it activates PFK, right? So it activates PFK, but it inhibits the phosphatase. It goes the other way. So you can see what happens here. If you have it, the phosphatase has a really tough time binding its substrate. This is the substrate of the phosphatase here. And if you don't have it, then PFK1 has a tough time binding its substrate. Right? If you don't have it, here's the substrate for PFK1 going this way. If you don't have this extra metabolite here, right, then the response of PFK to its actual substrate is much worse. Right? So this is telling you how it works. Any questions about that? So the first level of it is, okay, we have this extra metabolite, and it operates on the two enzymes reciprocally in these stages of the pathway. So then what controls this? Right? What controls F26BP concentration? And what controls that is another set of enzymes that are sort of parallel to the first two that are in glycolysis and gluconeogenesis directly. These two enzymes are sort of one step back, and their purpose is to synthesize and break down this regulatory metabolite. Okay, so here's those two enzymes. And those two enzymes turn out to be part of a covalent, in the same protein, basically, a covalently bound bifunctional enzyme here, right? Which itself can either be not phosphorylated or phosphorylated. Okay? If that bifunctional enzyme is not phosphorylated, then the portion of it that codes for PFK2 is active. And if that portion is active, then a lot of this metabolite gets made. And if a lot of this metabolite gets made, then glycolysis goes up because PFK1 is stimulated. Right? So what would stimulate, what would be a condition in the body that would make you want to stimulate glycolysis?
Insulin.
And that would be if there's a lot of glucose around. There's a lot of glucose in the blood, because then it's sensed from different organs. So insulin is a pancreatic hormone, a small protein, in fact, that operates through a cascade, and we'll be looking at the cascade a little bit later on, and it's also something you looked at last term. So that's what this dashed arrow means. It's some cascade of proteins going from outside the cell inside. The effect of it is to activate a phosphatase enzyme that dephosphorylates this bifunctional protein. When it's not phosphorylated anymore, then the portion of it that becomes active is the portion that makes this metabolite, which is also then going to activate glycolysis through PFK1. Right? And then thinking about it the other way, if this is phosphorylated, right, then the FBPase2 enzyme is active. And what that enzyme does is it destroys this. Right? So if you don't have this around, right, then you could ask, well, what's inhibited and what's active then? Right? If you don't have this around, then you've got more F26. Sorry. This is phosphorylated. Then FBPase2 is active. So if it's active, you're getting less of this and more of this. Right? So if there's less F26BP, right, then the gluconeogenesis enzyme is active. This is an activity trace of the gluconeogenesis enzyme. Right? When do you want gluconeogenesis to be active? Right? You want it to be active when there's low blood sugar. Especially you want it to be active in the liver, so that the liver can make glucose and ship it out to the bloodstream. That's what glucagon is telling the liver to do, basically. And the rest of it is the machinery by which it happens. Okay? So we have levels of control, right? And what that allows one to do is to have a finer level of